Okay, maybe we get started, then everyone will come in. So welcome back everyone for the third day of the school. Today we will start with a new set of lectures by Pablo Sartori. Pablo is a physicist and he is currently at the Gulbenkian Institute for Science. Gulbenkian Institute for Science in Portugal. Before that, he was a postdoc with Stan Leibler. Even before that, with John Howard at Yale. Did his PhD at the Max Planck Institute in Dresden. Looking very much forward to your talks, Pablo.
Can you hear me? Can everybody see me? Yeah, okay. I will welcome everybody. Thank you for coming. Third day, we're getting a bit in the mood, maybe a bit tired as well. I don't have I'm going to talk about something very, very simple. I'm not going to say very complicated things. Okay. This just fell. Yeah, I was just introduced, right? So I'm a theoretical physicist. I work in biology since essentially the beginnings of my career. I had a little stint at very theoretical physics, but decided that was not what I wanted to continue doing. And I worked on a diversity of topics throughout these years, ranging from bacterial signal transduction, chemotaxis, then biomechanics, and more recently, protein mechanics and self-assembly, as well as microbial growth. So it sounds a bit broad, but one theme covering all of these topics has to do with energy transduction. And I'm going to try to give you a flavor of how we think about this problem. Now, I would encourage all of you to interrupt me often. Okay, I really don't have too much to say, so if you don't do that, it's going to be a bit awkward. You know, finish probably 40 minutes and then last slide, nothing else to say. No? So please go ahead and ask questions. For some of you, everything will be very elementary. For others, maybe less so, but I'm trying to have everybody on the same boat, okay? And maybe before getting to bioenergetics per se, I will just say some things of how I like to think about biology and why I think energy transduction is an important concept here. So and before that, there's a small overview of the course, right? So today we're just going to be very, very mild. We're just going to introduce some notions of bioenergetics. Then, depending how we're doing with time, tomorrow, in principle, I will give perhaps one or two examples in which you can calculate energy dissipation or talk about bioenergetics of particular subcellular systems. And then the last day, again, depending how we're doing, how we did the other 2 days, we may do a bit more of a research talk, but also quite slow, so it's pedagogical, on the kind of things that I'm doing with Jonathan on microbial growth and the role of energy transduction there. Okay, so today's lecture only has two very simple topics. The first is I'm going to talk about 15 minutes of vague notions of why energy transduction may or may not be important in biology, right? So I think it's important to question yourself whether this actually makes sense. And the second is we're just going to go on the blackboard and do some very simple estimates of energetic quantities to get a feeling for how biological systems dissipate, how they compare to non-living systems, and how we could go about that estimating quantities, thermodynamic quantities that seem very inaccessible, okay? Is that more or less clear, the structure? Okay. Now, as I said, before talking about bioenergetics, I'd like to introduce an example of how I like to think about biology. And the way I like to think about biology is or the part of biology that I'm most interested in is the one that has these two concepts together of heterogeneity and functionality. And so these two ideas may seem very, very contradictory in some sense, but because heterogeneity normally is, you think of disordered systems, many components, that somehow it's messy. And functionality, we tend to think about something very, very structured. But when you look at something like this, right, you realize the heterogeneity and functionality, they come together very well, right? So this is a radio. For the youngest people in the audience, this is like an iPad. but for my generation, okay, it only had one or perhaps two buttons. And when you see this ancient iPad, what you notice is it has many different components, right? So it has about 20 or 30 different components, and each one is repeated a few times, right? So you have like 2 springs and maybe 5 cables, 1 magnet, right? And each of these components is special, has a purpose and a place to be. You cannot replace it by another one, maybe by another of the same kind, but if you put the magnet where the spring is, it doesn't work. So it's very heterogeneous. It's very peculiar in its heterogeneity, and at the same time, there's a very simple emergent function from it, as it just plays, music. Okay, and if you try to think about bioenergy, so we're going to talk a bit about radios. So you try to think about how energy may play a role in understanding a radio, right? Which then you realize that it's not really going to tell you the essence of how the radio works, the microscopic details of how the radio works, but it's more of a flavor that you can use to describe different aspects of the radio. So you can talk about the battery, right? And you can talk about the energy of the battery, and this is some kind of global energy budget for the cell, and associated to the transformation of the energy of the battery into other types of energy, there will be some dissipation. In this case, we tend to call it Hess law, and it's the radiation of heat associated to chemical reactions, right? Then you have wires that have a different role in the radio, that transmit information, right? And as they transmit information, you can also have the notion of voltage, but the wires will heat up as they transmit this information. And finally, you get to the speaker, right? The speaker vibrates, boom, 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 you hear. You can play it louder if you want. You had that option. And this will be dissipated somehow into friction of the moving parts. So you see, somehow energy transformations are important to understand the functionality, and dissipation is unavoidable. And one way in which you could try to think about all of this is that as the dissipation of the radio increases, a certain function of this radio, such as becoming louder, also increases. Okay. Now, of course, this cannot be. So these are the kind of things that one can expect of connecting energetics and biology. Whether this is a good explanation or not is up for debate, right? Because dissipation is not a tuning parameter, right? It's not a control parameter. You're going to change other aspects like the resistance or something like this, right? This is just some kind of global order variable, if you want, right? More than a control variable. And at the same time, we know that when we write function, this is a completely made up thing, right? It's whatever I want it to be. And that's, I want the radio to be loud, but other times maybe I want, okay, so the ordering of the slide is a bit bad. I'll just go like this, but other times maybe I want the radio to be quiet, right? And if I want the radio to be quiet, then the opposite will happen, right? So this is not, in some sense, will not be a universal relation because we don't know very well what to put on this axis, right? And it will be, but we can see last things about it, such as whether there's limits which are somehow hardware limited by the type of cables that we're using, right? Or whether there's some particular scaling here that we find relevant, okay? Is there any questions about how to think about radios? Yeah.
Something.
Okay, so I'll repeat the question for the people on Zoom or that didn't hear. So the question is this a bit of like a chicken and egg? You could think that as the function increases, the dissipation increases. Absolutely. This is, as I said, this is not a control variable. This is just something that will happen, right? And how you want to interpret this is up for debate. It's something that will happen, and then you can think of it a big deal, or think of it as a city byproduct, right? I think most people here would think it's a very interesting thing, but I would say this is something that we should all question a bit. Okay. And when I think about bioenergetics, the idea is a bit of whether we can translate this type of logic to biological systems, right? So when I tell you about radios, to what extent can we say these things for biological systems? And so I was walking the other day on the campus, and I saw there must be people doing string theory here, no? Because they have this diagrams of world sheets, very, very advanced stuff. So in string theory, they have the ADS-CFT correspondence. So I think we also ought to have a certain correspondence in biophysics, no? So we can have the radio E. coli correspondence, right? So the idea is that we can try to think of bacteria like we think of radios, right? Complex systems, many components, emergent ordered functions, right? And all of this happens with a high heterogeneity inside the structures. Okay. And then we can look at the little wires and cables and speakers that happen inside bacteria, right? And as in the case of radios, we will use different techniques, tools of physics, mathematics, and of course of biology, right, to address these different systems, right? So in our group, we work on different such problems, right? So for example, we work on the self-assembly of the cytoplasm. Cytoplasm has many different components, it's very heterogeneous, but it orders, right? You see things that are structured, like the ribosome, right? It has 100 components that come together. It's quite impressive. And we use some framework for that, but we also talk about dissipation in those systems, right? We also try to say something about protein structures, right? Very different type of problems, right? Very different type of tools. We analyze structural biology data, we look at mechanics and so on. And there, we also talk about energetics, right? We try to infer some kind of energy function for these changes. And also things like metabolism, right? Where functions such as growth of the whole cell emerge. You can also talk about energetics in this context, right? So you see very different mathematics, very different physics, different scales, but you can always talk about energy in this system. And I think that the key question that we need to ask ourselves, right, when we work in a problem, and we're interested in bioenergetics, is whether the type of constraints that thermodynamics will impose in the system of interest that we're working on are in fact relevant, right? Because we should keep in mind that thermodynamics was invented around then, right? For systems like this, and we're 200 and something years later applying it to systems like this, right? Of course, it's very universal and it's very flexible, but in the spirit of being self-critical, right? I think it's worth thinking whether a discipline that was invented 200 years ago will be the best answer for the kind of things we're trying to discover today, right? And of course, there's extensions of thermodynamics, recent extension of thermodynamics. Maybe that's the answer, maybe not. Okay, so in order for me not to be talking the whole time, I think it would be good if for each of you dedicates at least 1 minute to talk to the person that is next to you about whether your particular system you think is subject to constraints of thermodynamics. Can you do that for me? Can you talk to him and you to him about whether the systems that you're working on, you think it should be subject to the thermodynamics constraints are relevant for the systems of interest? Please talk a little bit, It's 1 minute, it's 60 seconds of your life. Otherwise, I'm just gonna be here talking the whole time, so.
Maybe you should clarify, what do you mean by that?
Yeah, what?
Yeah, is it,
Okay.
my system? Yes.
Yes, 100%.
My system?
Yes. I think you're all
Otherwise, why will you work together?
Yeah, why are we?
Well,
Well, maybe we figure out whether it's not relevant.
Okay, so that's 1 minute. Thank you, everybody. Now you know a little bit of what the person sitting next to you is working on, and we all relaxed a little bit, and I'm sure you'll told each other, yes, thermodynamics is very important for what you're doing. I guess it's a bit self-fulfilling prophecy since this conference about the bioenergetics, but it's worth at least dedicating 30 seconds to think about it. Okay. So I'm gonna move to do some simple bioenergetics estimates, okay? And I'm gonna fill probably 45 minutes with that. We'll do a little break maybe after 15 minutes or something, because 1 hour and a half, I cannot talk for so long without boring myself. So I'm just gonna, before that, I'm just gonna have a 2 slides overview of thermodynamics, just some things that I find interesting. I won't give you a formal introduction. I think maybe Massimiliano next week will take care of, at least for one part of thermodynamics, giving a very structured view in 2 days, which I'm sure will be great, that I will miss. So I want to talk a bit about the one minute about the prehistory of thermodynamics, of the old spirit. And one of the old examples of how people were doing thermodynamics is Boyle's law that you may recognize as this. And I just want to show you this original printing from the measurements that Robert Boyle, right, did to convince himself that indeed pressure was inversely proportional to volume. And this was a quite cool experiment. He had to build a like 3 or 4 meters high mercury column. The interesting thing in this experiment, I mean, these are just little fun historical facts, is that in order to compare the experiments and theory, you need to look at these 2 columns side by side, okay? And there's 2 things to notice. First, that the numbers don't have point notation because it didn't exist at the time. And the other is that you may wonder, well, why didn't he just use a plot? Right? And the reason is that although this is post the treatise from Descartes, is very little after, and it was still not very much adopted. No? So this is how old the origins of thermodynamics is. Okay, this is like 370 years ago, 360 years ago. But if you plot the old data from Boyle today, this is the kind of things that you get. Right? So in some sense, this is the spirit of thermodynamics, right, which was trying to find simple phenomenological relations among microscopic variables of systems, of non-living systems, right? And that's one way to try to do thermodynamics about biological systems, which I will not talk about, right? Which has to do with maybe not about energy, but maybe something like the growth laws that Terry Hua is doing, right? Where, okay, it's not really thermodynamics, but it's the spirit of thermodynamics, a simple corresponding description of biological system and finding relationships among them, even if we don't know where these relations come from. Okay, and then the second period, or the next 2 steps, I would say, in thermodynamics, were when people were developing a steam engine such as this one, right? So this is a design of a steam engine from James Watt. It was at the time very efficient. So James Watt was one of the pioneers of using this little thing there, which is called the governor, and this allowed to control very well the steam engines and make them improve their efficiency. But people still didn't know the limits to how to construct steam engines. Right? They didn't know how good can a steam engine be, or how efficient we can make it. And then there was this young hero, right, C.D. Carnot, who said that essentially we should, in a quite verbose way, as it was customary at the time, that we should abstract this kind of all engines. We don't care about all these details. We just should think about them as something like this, where you have high temperature, low temperature, an engine, and some work that is performed, right? And once this leap of abstraction was done, well, there was the writings were forgotten and so on, but the formalism of thermodynamics was developed very fast, right? So somehow we need this kind of leaps. And today, it's popular culture even know that these 2 claims, no, entropy increases and energy is conserved. Okay, so this is more or less a very rough overview of the history of thermodynamics. So in the any comments about the history? It's a bit. So some people, I would just say, so C.D. Carnot actually, I don't think he wrote, I only know 1 paper or treatise by him, right? So he may have an H index of 2, just for the younger audience that worries about this, right? But he changed the history, right? He changed the history of thermodynamics and therefore of energy transaction, of the economy, capitalism. And maybe because this was fueling capitalism, right, at the time. So maybe we consider him a hero. As I remember, there was an old student of Massimiliano had a picture of him always hanging on his door. Okay, so what I will do next, I'm just going to give you some estimates, some of the ones that Jonathan went already over, but I'll repeat them again with a bit more care on the blackboard, because I think it's pedagogical to see them. And I'm going to try to say something about the first law, right? So some energy of living systems. And I'm going to try to say something about the second law, so dissipation. There's a question, Vijay.
No, sorry, a comment. I just want to comment that looking at the dates, so we understood thermodynamics well before we understood matter was made up of atoms.
Yes.
So phenomenologically, right? So maybe I mean it's a lesson to be learned in biology. Don't just go after the molecules.
Oh
but
yeah,
maybe-
right. So there's, yeah, I feel like I'm saying this kind of things all the time, so maybe I should. But yeah, so this is something that, in some sense, I guess since the physics audience, so for us it comes quite natural. Oh, what happened? So for us it comes quite natural that to have coarse-grained theories, right? So in physics we know that a theory is something effective, right? At a certain scale. So almost everything that matters at the cellular scale has to do with electromagnetism in some sense, right? Because it's not gravity and it's not strong force, right? So it's some different flavors of electromagnetism, right? But nobody will get obsessed about talking about photons exchanges, right? So in what Vijay is saying is that you can have a very good understanding of a system without understanding the microscopics of it, right? I mean, that's how, well, you know, many physical theories were built. That's how somebody was talking about the normalization group, about fluid dynamics, right? So Navier-Stokes equation, I think it's an almost better example, right? So to understand the fluid, right, to understand how water flows, and to understand turbulence, you need to write some equations for a fluid. And these are the Navier-Stokes equations. Nobody cares how these equations are derived from microscopic origin. In fact, formally, you cannot do it because the microscopics will be reversible, Navier-Stokes is irreversible. So mathematically, we cannot properly do it, but we don't care. You can, and in fact, you probably wouldn't be able to understand this emergent phenomena if you follow track of everything.
Just to provoke a little bit,
Yes.
maybe I make the counterpoint, which is, of course, biology maybe have started off this way, somewhere along lost its way into getting into molecular details. And
sure.
Maybe it's coming, but because it has made that detour, now we know that molecules do play an important role.
Yes.
So I don't think we should.
No, that's true. So now let's talk about the biology side. So the other says that biology is not physics, right? So physics has this very clear separation of scales often, and of time and space. You can really just do this coarse graining very happily. But I think that the magic of biology is that it plays a bit with both, right? So in some sense, you have these effective descriptions that seem to work, right? But in another sense, you then perturb a very small thing and you disrupt the whole function. This doesn't happen in systems like this. Well, maybe in systems like this, right? But it doesn't happen in standard Navier-Stokes type of equations. Okay. Yeah, no, but it's a very good point. Any more comments on complete silence? Okay. Okay. So maybe now would be a good time to lift this thing, and I'll just go over some of the estimates that I told you about. And the idea here is to give you a feeling today, not only of the results of these estimates, but also on how to do them and the kind of fun questions you can try to answer yourself. So that's very big. Maybe I can start here. That's probably enough. Okay, so Jonathan started, I don't know if maybe the first thing he said, but one of the first things he said is this very beautiful notion that humans dissipate more than the sun, right? Which seems shocking, so I thought we can go over it again together. And for that, so the idea is calculating how much heat humans and the sun radiate, right? And compare it. So we can start by, so let's say, right? So the first thing we need to know is what is the mass of a human, right? So how much do I weigh? That's the first question. What? How much is my weight in kilogram? My weight, yeah.
70.
70, okay. Other guess? How much do you think I weigh? 75, okay. How much do you think I weigh? 75, okay. I personally have no idea. I haven't weighed myself, I think, since I'm 17, but it seems 75 is a good number, right? So okay, let's say, so the mass of a person is about, so if you say 75, I'm going to write 100. Okay, so I weigh 100 kilos, right? Is 100 kilograms. And the other thing that we can ask is, how many calories does a person consume per day? So do you have any idea of these kind of numbers? So for example, Alberto, right? About 2,000. Does everybody agree with that number? That's a lower limit. Oh, 2,500. Okay, so that's very precise. So if you don't, so here's so I have no idea, but you can do a very simple trick, right? Where you don't really know these numbers. You want to give a rough estimate. You can do a geometric mean, right? So I think everybody agrees that it's probably less than 100, more than 100 calories, right? And probably less than 100,000 calories, right? Because that sounds like a very crazy number of calories to consume. So what you can do is you take the geometric mean, right, and this is in kilocalories, and the number you will get is something about 3,000. Okay, so this is an example of how to do we don't know anything about caloric content. You can still say something, okay? Just something to keep in mind. Where? Oh, 3,000, sorry, thank you. Okay, so then we need a little conversion factors, which, so one conversion factor is, where do I put them? I can put here, I'll put the trick. So one is does anybody know how many joules a kilocalorie is? This is a very funny thing. So a calorie is about, a kilocalorie is about 4 kilojoules. So somehow we felt the need to introduce a new unit just for a factor of 4, which puzzles me. And the other thing that you should remember is that one day is this we all know, right? It's like 60 times 60 times 24. And that's about 10 to the 5 seconds. Okay. And so if you put this together, you can say the power dissipated by person is so what, 3,000 times 4,000 times 10 to the 5 seconds. Did we make a mistake? 3,004. Oh, 4, sorry. Yeah, it's joules, Sorry, I'm writing joules. It's about 100 watts. So 100 watts is about a lamp, a bulb of the ones that were not LED, right? So that explains why there's a few people in the room, it gets hot. Okay, and we can calculate little p per person, right? So per kilogram would be about 1 watt per kilogram. And that's the quantity that Jonathan was mentioning. Is it clear? And what Jonathan was doing was comparing it to the sun, right? And for the sun, these things have been measured. So we know that the power dissipated by the sun is about 4 times 10 to the 26 watts. And the mass of the sun is about 2 times 10 to the 30 kilograms. And then if you divide these two, right, you have about 10 to the minus 4, should put always approximated, 10 to the minus 4 watts per kilogram. Okay, so when I saw this number, I mean, I was very puzzled, right? Because this somehow intuitively doesn't seem to make sense, right? And the first thing I thought is, oh, maybe it's just about the core, right? So the core of the sun, really where actual nuclear reactions happen, is very small, but it is not so small. So that kills 1 order of magnitude, right? So it's true that the sun has a core where the nuclear reactions happen, but that weighs 10% of the total mass. So that's still a big gap, right? And what you can do, but I won't do, but if you want, if you've never used, so how many biologists are there in the audience? You will have a few. You all probably have heard of this formula, right? You all seen this formula, right? But have you ever used it? No. The biologists, have you ever used it for something? So if you want, you can actually use this formula to understand how many kilos the sun is turning over, right? It's a very simple calculation that you can do. And essentially what happens is that the sun has nuclear reactions, right? Nuclear fission. And as it does, it loses a bit of mass. And this mass is converted into energy with Einstein's formula. And then you can see that what happens, essentially, the sun is turning out very little of its mass per unit time. So most of the reaction of the hydrogen that it has is not reacting. Okay, so that's about the sun. So we found that the conclusion is that per kilogram, a person radiates much more than the sun. The next thing we want to try to estimate is talk about, so this is about the first law. We want to try to do some estimates about the second law. So we'd like to say something about entropy production in the Earth, right? In the whole Earth, and then try to compare that to the biosphere. So we know how much a person dissipates. We know that it's more. Yes, you have a question.
Statement about like efficiency, or what should I be thinking about? Why is this so? Like, I think that the sun isn't doing like each per kilogram of the sun isn't producing as much energy. What should I be thinking about?
So in some sense, there's a bit of a populist statement, right? But essentially, what the claim here is that the amount of heat that a person is radiating per kilogram is much higher than the amount of heat that a kilogram of sun is radiating. And the reason why it seems surprising is because the sun looks very hot, right? Now, I don't know if there's so I'm still trying to understand whether there's a profound reason for it or not, right? So my first guess is maybe there's a trivial explanation for this, or maybe it's just that in some sense the sun is not so efficient, so it's not as reactive as intuitively it may seem, right? So a lot of the mass is in fact not undergoing nuclear fission, right? Is there any more questions, commentaries, Vijay?
Maybe it's a naive thing. So I don't understand. So 3,000 kilocals is the energy consumed by a person.
Right. So
but the 10 to the 26 is the energy outputted by the sun.
Yes. So
I don't know how
this is very good point. So I didn't say that. The assumption here is that we're eating somehow all the food the beginning of the day. At the end of the day, we're very hungry, and somehow all the energy has been dissipated, or that we're in a steady state, and that's the amount of energy that we're consuming, we're somehow outputting as well.
Yeah,
and
but
we're not getting.
You're probably
fat.
using some fraction of that to maintain the structure as well.
You need to have some fraction to maintain the structure.
I'm saying to maintain a structure, it's not a maximum entropy state. So you would have used some part of it to reduce your entropy.
So you use it, but you won't accumulate it. I think that you're not growing. So ultimately, everything, I mean, different ways, right? Of course, not everything heat. And on our last day, we'll say something about that for microbes, right? But the idea is that you're not keeping the energy, it's just somehow going away, right? We're going to assume this is going away as heat. You're a black body in some sense. Yeah. Yes.
One quick question. We write, so
This is a very good point. You should do the calculation and send it on the mailing list. You know, I haven't done it, but we're going to talk a little bit about black body radiation next, but not about the human. Right? But any more comments, questions, Yata? Well, I mean, you can. So the question is, can you somehow say, well, we dissipate more because we're more functional than the sun? Sure. I mean, you know, for a pop talk, you can say things like that. You know, I wouldn't write on a paper. Okay. So yeah, more comments. Yeah. Can you please talk a bit louder? I mean, with the microphone would be great. Otherwise, I'll repeat the question.
Hello. Yeah. So if a human does not eat as much as what's eaten normally, will that person dissipate the same as the one who is consuming around 3,000 calories? So the question is basically, does it depend on the food that the human is consuming, or it depends on, I mean, you know, stored food and the dissipation, I mean, the breakdown by the cells?
So this is a very good question for which I don't know the answer. Maybe Jonathan even knows it a bit better, but so yes, you can lose weight if you're not eating enough, but there is true that there may be people that need to sustain their own weight, have a lesser caloric, you know, a lower caloric intake, and there's also people that can fast and yet stay somehow adjust their metabolic rate, no? So for extended periods, and sometimes during disease we do things like this, and we do self fasting, right? We're going to do anorexic state to lower somehow. I mean, one of the ideas is that we fast to lower our basal metabolic rate to fight infection and things like that. So I think the metabolic rate, I'm not an expert on this, but I think it's probably adjustable.
Right. So do they dissipate similar? Like,
I would say no, because you can dissipate, you can adjust. By metabolic rate is how much you dissipate, so you can adjust that, right. Change the heartbeat, the respiration, I guess, and all that.
Right. So there's just a follow up on that. So there's also cases where people eat the similar, you know, amount of food, but somebody tends to digest it fast, and then, you know, they get hungry faster, and the others don't get hungry as much.
Yes.
Does that also got to do with, you know, how much it is dissipating, and
I really don't
how
know.
does it happen? I mean,
I really don't know. So I'm not an expert in human physiology by any means, but I think it's a very interesting question. I think the energetics of human physiology, I don't know how well understood they are. I don't know how seriously people have taken this. They're human calorimeters. I think Francesco had maybe
Okay, so I was wondering, like, those 3,000 kilocalories, right? They are the input of energy.
Yes.
But then part of that energy might be used for something useful. So maybe the dissipation is less than that?
So this is a good point, right? So like a good thermodynamics says, so in fact, what do we mean by dissipation, right? So in some sense, when I say that all of that will be dissipated, is as if you were just going to move on air, and therefore everything will be dissipated into the environment as heat. By something useful, do you mean like I'm pushing a spring or something?
No, more like, I don't know, maintaining all the processes in your cells, et cetera, et cetera, et cetera.
So what I'm trying to say is, like, if I'm always being very vague, right? But if everything is at a steady state, then you can assume that what you're eating, you're eventually going to dissipate it if your body stays essentially in the same state. So yes, you will use this energy to do things, but all these reactions will be dissipating energy, and in the end, the state.
But you waste also molecules, right? You don't dissipate only heat.
Yes, yeah, this I said, right? So of course you pee and you do things like that, right? So part of your energy goes out of you in other manners that is not heat. But that's I would say that's dissipated as fluid friction, right? The things that are moving. Yes. Yes. Some. Mm-hmm. Yes, so I guess, I mean, maybe let's not get, so yeah, some fraction is radiated, but what I'm trying to say is that, yeah, just say some is being moved and not moved, but eventually it should be dissipated in terms of friction. Yes. Mm-hmm. And is it the mechanical work eventually transmitted to heating the Mm-hmm. Mm-hmm. Okay. So right. Okay. So the comment is that essentially I'm not taking into account the work that is actually done by the system, right? I'll take all the criticism. This is a very rough estimate.
So Pablo, just as a follow-up to all of this discussion,
yes.
There must be a measurement of the total heat that's being released from a human, right?
Yes.
And
it's about that.
It is about
that.
Yeah. So that answers everybody's question, I think.
So
it's
that,
not
right?
a fraction of that, is what you're saying, but it's really.
It's about that, right? So people measure it by measuring the oxygen consumption rate, and they have a, it's about that, and human calorimeters and so on, right?
So I want to do the
Okay. They get the same number. Okay. So maybe let's move on a little bit. So, okay, last question, and maybe move on a little bit. Yeah. Okay, so it seems to be and final question before we move on to even more populist estimates.
So you've done this calculation for two different scales, like one is sun, that's huge, one is for human. Question is, if we go down to the level of a cell, do something similar, some similar calculation for a cell. First, we'll just consider the number of cells that a human is made up of. Is that directly how we go down to that level? Or we do a separate calculation for how much energy a cell consumes, calories of cell, energy consumed, and the mass of a cell. And intuitively, can we say that it will be more than this 1 watt per kg?
1 watt per?
The calculation for sun comes out to be 10 to the power minus 4 watt per kg. For human, it is 1 watt per kg.
So how does it apply to cells?
Intuitively, should it be more than this number?
So the question is, can we just scale this down to cells, right? And what I say to that is there's a whole discipline on, so let me say two things. First, we will talk more about cells in 2 days, right? So we've done this, and what turns out is the numbers are higher. Second, there's a whole field of study, if you want, about how, or subfield, how metabolic rates scale with system sizes. Okay, and it's not clear that it's linear, right? So there's a whole, maybe you have heard about, or if not, you can Google Kleiber's law. Right, so the idea is that the metabolic rate of organisms of different sizes doesn't scale proportionally to their mass, but there's some sublinear scaling. So, okay. All right, so if you thought that was rough, now let's go to this other very fine estimate that people do. which has to do with how much energy reaches the Earth. And then we can even do a crude estimate of the entropy production of the Earth by looking at the difference between the photons that come in and the ones that come out. Okay, so now let's see. So how much energy reaches the Earth from the sun? Okay, so for that, we're gonna first calculate, we know how much energy the sun is radiating, which is here, right? So we just need to divide it on a big shell that reaches the Earth, right? So what we're gonna assume is this is the sun, and this is the Earth. And well, the Earth has a certain radius, we're gonna call our Earth, and there's a certain distance, which you can call the distance to the sun. And these numbers are roughly known. So this one is 1.5 10 to 11 meters. And this is a few thousand kilometers. Let me see exactly, so 6,000 kilometers. So 10 to the 6 meters again. Not so big. And so what we can do is we can say that the power that is arriving to the Earth, let's just call it like that, is the power that the sun is radiating over the distance, right? A sphere. So that would be the density. And if you do this, you get something like 10 to the 3 watts per square meter. Okay, it's a fun number, right? So that means that, of course, you know, there's a lot dissipated by the atmosphere, but roughly per square meter, no? So if you have a person here, right? More or less the projected area is probably about 1 square meter, right? So you're getting about 1,000 watts, and you're radiating as a black body based on metabolic rate and very crude estimates, about 100. 10%. It's funny. Now, what happens? One estimate that sometimes you will see that is very fun is that if you have here the sun is radiating to the Earth in the form of photons, right? And the Earth is also radiating in the terms of photons at the galaxy scale, not the solar system scale. But the type of photons that the sun radiates are different. But of course, the type of photons are the same. But the temperature of the sun is different from the temperature of the Earth. And what you can do is, again, assume that the power that is going in the Earth is equal to the power that is going out of the Earth. Okay. Am I using dots? Okay. And yes, you can get the temp, and you can also get the temperature of the Earth as a black body, which you just calculate for humans, right? So for as a black body temperature of the Earth, well, you can calculate it actually, but you can also just put something reasonable, like let's put about 300 Kelvin, and the temperature of the sun is about 6,000 Kelvin. Okay. And so if you assume that all the power that is coming in the Earth eventually goes out of the Earth, which is assuming that the Earth is at a steady state, okay, meaning there's no energy that is coming in and being accumulated in tectonic plates and whatever at a certain scale, you can then try to calculate the change in entropy by the entropy that went in the Earth and the entropy that went out of the Earth. And for that, there's this this nice formula, which is the entropy of a photon gas that you may or may not remember. So this is one of those beautiful statistical physics calculations that you do in university. I think that the other very beautiful one is the balance for a neutron star, which I think Chandrasekhar did. No? The balance for a neutron star. You know, when you do the right. This is the other very beautiful. I remember when we did that. Okay, so if you do this and you assume that U in equals the U out, then you can say that the change in entropy should be, you can even say that the entropy production, it should be about 4/3 of this power times 1 over this minus 1 over times the sun, right? Because the temperature of the sun is bigger, this will be positive. Okay. And because in fact this is very small, you can just, well, you can plug the numbers that we had there, and more or less what you will get is something like this. That's per Kelvin. Okay, so that's a number. Is this very crude estimate also more or less clear? Right, so we're just doing an entropy balance. We're just saying there's the same amount of energy coming in the Earth like going out of the Earth. So for that, we're assuming that the Earth is at a steady state, right? There's no energy being accumulated in the Earth. And so that we can calculate how much entropy is going in, how much entropy is going out. We do the entropy balance, and then we say the difference is the entropy production. Is a P in, I guess. Yes, but multiply by the surface. Oh, I didn't. Yeah, I forgot to write that. I have it somewhere. Multiply by the surface. Yes. Yes, my notation is not. I've been super careful with that. Okay. And if you multiply this by the temperature of the Earth, we very roughly say that that will give you a scale of the free energy dissipated. As you do TS dot, what you will get is something about 10 to the 17 watts, right? Because we said it's the temperature is about 300 Kelvin. And one thing that, so the reason behind this number is varied, right? So there's many factors that contribute to it. Probably the main one is scattering in the atmosphere, right? So a lot of scattering. The same calculation you can do for other planets, right? So obviously this has nothing to do with biology. But what you can do is you can try to estimate how much, if this was done by biomass, how much would that biomass have to dissipate? Okay, so how does the biomass dissipation estimates that we compared before would compare to this global number, right? And now to do that, you will need to have an estimate of how much biomass there is. So how many kilos of cows and people and bacteria there are in the planet. And luckily, there's one very beautiful paper in which they've done this very, very carefully by Rob Phillips and Ron Milo. And is it also the person you're working with in that paper? No, no. Okay, well, some other people. I don't know. And what they estimated, they gave quite a precise estimate. They say like the mass of the biomass is about 150 times 10 to the 12 kilograms of carbon, which is about 6 times 10 to the 14 kilograms, carbon kilograms, okay? Okay, so then you can divide this by this, right? And what you get is a by the mass of biomass. You get something like, and you assume that only about 20% of the biomass is made of carbon, right? Because carbon is not the dominant factor. So what you get is something like 50 watts per kilogram. Okay. So what does that tell us? So that was a weird estimate. Essentially, what that tells us is that if we compare the dissipation that we estimate at the Earth, looking at from outside, to the dissipation of the biosphere in this very, very, very crude way, right? The numbers that you get, of course, is that the dissipation of the biosphere is much, much smaller, but not like orders of, not like 3 orders of magnitude smaller. So I found that quite shocking, right? So that means that perhaps we're even in the few percent of contribution to the dissipation of the planet, right? So if you take into account, of course, most of it is photons get scattered in the atmosphere and so on. But if you take into account how much we all together radiate, okay, it's almost at the planetary scale. Okay. Any questions, comments about this part? Yeah. I think somebody may be bringing the microphone.
Is it working? Okay. Sorry, could you possibly just explain again these 3 figures? I didn't really understand what they.
Right. So what we did is so let's go over this again, right? So what we did is we used a trick that sometimes is done in astronomy to talk about entropy production, right? In which you calculate the difference. So this is more or less clear, the difference between the entropy coming in and the entropy going out, just assuming that the Earth and the Sun are black bodies, right? So very, very simple. And we get a number for how much dissipation happens in the Earth, how much entropy production happens in this process, multiply by temperature to have something like a free energy term. And the logic of these last 2 steps is the following, right? So we know this dissipation doesn't arise predominantly from the biosphere, right? This is not animals dissipating to the Earth, right? The Earth is it's a planet and it has an atmosphere. And so when photons come in, they get scattered, right? And that produces entropy. But we can try to estimate if you divide that dissipation per the total amount of biomass in the planet, how much would the biomass have to dissipate? Does that make sense? Right?
Yes, yeah.
And the number you will get will be, it is, as it should, be bigger than what animals actually dissipate. The question is how much bigger, right? How much bigger? So what this is telling you compared to so you should compare this to the 1 watt per kilogram, right? So what this is telling you is that if humans would have to dissipate to produce the whole dissipation in the atmosphere, they would have to dissipate much more, but not 3 orders of magnitude more, maybe just 1 or 2.
Is there too much focus placed on humans? Because what about all the plants and algae and bacteria?
Right. So that's a good point. What about all the so this is, of course, this is a very rough estimate. I insist on this, but this is an account of the total amount of biomass. Okay. What we don't know, what we haven't done, and I would love to do if somebody wants to do it with me, is in this total account of biomass, there's very different types of organisms. Humans are, of course, a very small part, right? There's bacteria, there's fishes, there's archaea, there's plants that do photosynthesis, very different, right? Then there's mammalians and so on. Each of them per kilogram will dissipate something different, right? Here we're just doing a brutal approximation and assuming that they're all human, right? We're all human. Every kilo of biomass dissipates the same as a human. I mean, we know that's not the case. And in fact, it's probably a very crude estimate because human is absolutely not representative of the dissipation of organisms, right? Because they scale across scales because of Kleiber's law and so on, right?
But this is suggesting that the average energy usage per kilogram in a kilogram of biomass is higher in non-humans than humans are relatively small in their entropy.
Okay, so two parts. I mean, you may think of that because this number doesn't match, but in reality, this number shouldn't match with the total. So you wouldn't expect that the dissipation of the Earth matches the dissipation of the biosphere, right? This shouldn't be the case because there's dissipation for many other reasons, non-living reasons, right? But it's also true that humans are not representative, are not very representative, right? Vijay, yeah.
Can I turn the argument around and say that if there were life forms, this is the upper limit that they should dissipate? All dissipated. If it's all,
yes,
it's all
yes.
dissipated.
For example, that could be a way to think about it.
So there can't be life forms which are dissipating more than this.
Right. So you're saying there couldn't be, no, because it's heterogeneous, right? Different organisms. You can have.
No, on the average.
You know,
on the
average.
You put in some number, 1 watt per kilogram. Some assume it's a homogeneous distribution. I'm saying if there are life forms on a planet, then this is an upper bound on.
Right. But I think this would be a very interesting. So this is one of the things that I would like to do. So if somebody wants to do it with me, because it's very boring to do this alone, right? But we have a very good notion by now of the distribution of this. Okay, so that people have done very good research or knowing how the biosphere is distributed into different compartments of different types of organisms, to which presumably we can ascribe different metabolic rates, right? And so you can, I think it's definitely possible and not crazy tedious, let's say, do a global assessment of biosphere dissipation. This can be done. You just need to be patient. So somebody, you know, yes, anymore.
Yeah, one more thing I would like, it's a very major assumption that Earth is at steady state at this case.
So the question is, yeah.
So my question is basically like when we are actually looking at figures like much larger than this, we have to also associate the fact that we have assumed that Earth is at steady state, which means that you are assuming that all the energy that is coming from the sun is being consumed by all the biomass and is being dissipated at the same rate as well. Like, which is a very bold assumption to make altogether.
Or scattered,
yes,
right?
That all the biomass is consuming all the energy that Earth is getting from the sun and then dissipating all of that.
Not the biomass, the
yes,
Earth. The Earth.
Yeah, Earth's biomass. Overall.
Earth.
Yes.
Earth. It can also just be scattered by
that is a very major assumption.
Yes. So the question, the comment is, we're assuming that the Earth is a steady state. That's a major assumption. It's absolutely true, right? So one will have to carefully look at the geology of the Earth and see to what level this is a fair assumption. Right? Maybe for I don't know. I mean, you know, I looked at a couple of so I prepare all this myself. So I look at a few astronomy papers. I tend to say it's a reasonable assumption, but I haven't looked too much into it, to be honest.
Well, I had a question. So when it comes to this energy per mass, like what would you expect? Would bacteria dissipate more energy for their mass, or would higher organisms be dissipating more?
That's your question?
Yeah.
Yeah. So the question is, do you expect that bacteria dissipate more or higher organisms? So of course the question is per kilogram or per gram. Well, I don't know what my expectation would be before knowing the answer. I can tell you the answer. So the answer seems to be that bacteria per kilogram dissipate more, substantially more than higher organisms. Okay, so essentially bacteria is sort of just energy going in and out and very little staying in the next bacteria, right? So the energy content of biomass in some sense is relatively small, but the flows of energy are very large. Just one more question and then 20 minutes, okay.
Yeah, so I got a little lost in the previous part when you said that about the entropy balance. So can you please explain how that happens?
So what we're doing is we're calculating the entropy of this and the entropy of this. Right, so the sun is radiating energy, right? Heat, let's say, but heat is photons. It's being radiated as photons. And so what you have is a gas of photons is hitting the Earth. That's what's coming in, a gas of photons. Now, for a gas of photons, you can do thermodynamics of a gas of photons. You can do statistical physics and get an equation of state for a gas of photons, and then you can relate the energy of that gas to the entropy. Right? And that's what we wrote here. And we're doing the same for the photons radiated from the Earth. So the Earth, if you look at it from a spaceship, it's also has a spectrum, right, of radiation. And we're approximating those spectrum to being, black body spectrums, and then we're saying that each of these gases of photons has a certain temperature, right? And the formula tells us that the entropy of a gas of photons is related, the relation between the energy and the entropy has to do with this temperature factor in the denominator. And we're assuming that the energy that is coming in the Earth is the same that is going out of the Earth, and that the Earth is at a steady state in terms of energy. And so the only difference is the energy that the photons, so the energy of the photons coming in is the same as the energy of the photons coming out. But you could say the photons at different temperature, so they contain different entropy. And so we're taking the difference between those two, and we're using that as a proxy for entropy production in the process of coming in and out.
Okay.
Okay, so I wanted to do a break, but now it's only 17 minutes. We can still do a 1-minute break, if that's okay. While I prepare my next and last rough estimate. Should we do that? Is that okay? Okay. 1 minute. The final thing I'm going to do, this is not due to me, this is, we have 12.5 minutes, maybe it's enough, so I'm gonna erase this. The final thing has to do with a brief, I touched upon it, and due to a question. So thank you very much for asking questions. And has to do with the entropy of biomass. Okay, so far we've talked about how much energy a human consumes, and we've also estimated the entropy production of the planet, and we've seen that it's much higher than what you would expect from the biosphere, right? And I've made a call for help if somebody wants to try to calculate it carefully, the entropy production of the biosphere by using metabolic rate and the mass distribution among different species. And one thing that we, maybe I shouldn't have erased everything actually, but one thing we could compare all of this to is with the actual entropy content of biomass, right? So you see, biomass is like more or less like any other system, right? So you can burn it, right? You can burn it, and then it will release heat, right? So it has a certain energy content, a certain entropy content, and it has a certain enthalpy content, right? And it seems if you're interested in bioenergetics, you're interested in energetics of bio things, it would be important to know the thermodynamic properties of those bio things, right? And the type of, temperature is important. We know the temperature of a human. We know the pressure inside cells, but it will also be good to know the energy and the free energy and the enthalpy and the entropy. Now, these numbers are hard to measure. Not that hard, I think. They're definitely not very popular experiments, right? But people have done some. So the enthalpy of biomass is done essentially by combustion, right? So you burn biomass. You take let's be gentle. You take an apple, and you burn it, right? And then you see how much heat is released, and then you try to, you know that when this combustion reaction happens, the difference between the enthalpy of reactants and products should be equal to the heat release, right? And if you know the properties of the reactants, except biomass and the products, and you measure how much of each thing is and how much heat is released, the difference should be the enthalpy of biomass. Right? So this is something people do. Yes. What is entropy in biology? So this is a very good question. I don't know to what extent that I can give you a satisfactory answer, but maybe the best way to think about it is that one shouldn't be too scared of the word entropy, right? Because it's, do we know what is energy in biology? I mean, it's just another similar quantity to energy, let's say, that seems we're a bit less familiar with, right? So when you talk about energy, if you ask yourself, but what is really energy? I mean, you won't have a good answer, right? It's just something that you're used to work with, right? And you should think that it's just a material property, right? So if you have a kilo of water, right? This kilo of water will be at a certain temperature, certain pressure, of a certain energy, and we have certain entropy. No different, right? So you say, what is the energy of water? What does it mean, right? You don't know. And then you say, oh, you say, well, depending on the conditions or whatever, the free energy is the capacity to do work or something like that, right? Because of some law of thermodynamics that tells you difference in free energy is equal to work, right? So entropy, I would think about it in that way. It's just an extensive property of systems that we can measure with some experiments, and that we can, yeah. Okay, so the question is, how do you measure entropy, right, of biomass? So that is an experimental question, right? So what would you do to, in fact, measure the entropy of biomass? So people have measured, a few people that have attempted to measure entropy I don't know if, you guys still here? Yeah. Entropy of biomass, and the way they have attempted to do so is by doing low temperature calorimetry, right? So essentially, you use the heat cap. So you measure it the same way that you would measure it for an inert material. So what you do is, I think you dry biomass, right? So you take biomass, you remove the water. Sorry, let's forget about that. You dry it, you compress it, probably you chop it into, and then you say, well, now I have, this is my kilo of biomass, right? And I'm going to cool it down very much. And then you heat it up, right? And then you measure little by little the heat capacity. You know? So how much heat it releases, and that gives you the heat capacity. And then you know that the entropy is integral of the heat capacity under certain conditions. No, there's some relation like this, probably. And that's how you measure it for standard materials, and that's how people have measured it for biomass. People have actually done that, and they got a number. Now you can say, well, but that's not biomass. I mean, that's like dried and whatever. That's true, right? So then you say, okay, so how do I do it for the other thing? You can, and people, what they do is they estimate how much entropy happens due to hydration, so to interactions with water molecules and so on, right? And well, I only have 6 minutes left, so I won't have time to go over this beautiful estimate. So maybe I'll just give you a flavor of how it's done, right? So this is an estimate done by a gentleman. I don't know what his background was. His name was Harold Morovitz. He has a very nice book on. You smile because he's a peculiar. I mean, he was one of these people that had a very holistic view on metabolism and understood very well thermodynamics, and he could do very
Don't worry about going a few minutes.
Okay.
You followed by poster session.
Okay. Well, but okay, if you say so. Okay. And and he knew thermodynamics, and so he was sort of, unlike me, he had the unique capacity of understanding very well the field of metabolism, very well the field of thermodynamics, and somehow bridge between the two. But he was a bit obscure to read his articles and his textbooks. So it's a bit, I don't know, it must be, I mean, So in one of his textbooks, there's a chapter where he talks about all of this entropy of biomass, how do you estimate it, and so on. A very difficult chapter. I've read it many times. Sometimes I think I understand it. But then he also has a little paper, less known, a very short paper, in which he does the crudest estimate of entropy you can imagine. And as is always the case for him, you get roughly the right number. So this is the crude estimate that he does, right? So he says, this is a bacteria. And a bacteria is made of proteins and RNA and DNA and lipids, all these things. We say, okay, whatever. For sure, it has atoms, right? So it has carbon, hydrogen, oxygen, nitrogen, phosphorus, sulfur. Okay, so he even goes all the way there. So let's just say that a bacteria, it's so when you say the spherical count approximation, right? So this is the sort of the cubic bacteria approximation. Let's just say a bacteria is a lattice of a lattice of atoms. Okay, and so in each of these lattice points, we put here there's a carbon. I mean, I cannot really draw in 3 dimensions, but like down there could be a hydrogen. Here's an oxygen. Maybe here's a nitrogen. Okay. And he even says the coordination number is 6. So this is an actual cubic lattice, right? So each of these little cubes can interact with the one above, with the one to the one side, to the front, to the other side, or in the back. Okay, and they will interact via chemical bonds, right? So you may have, if you cut one sheet of this, you may have things like this, like hydrogen, hydrogen, oxygen, oxygen, carbon, hydrogen, hydrogen, carbon, hydrogen, I guess. No, and then he says, okay, so you can have things like this carbon has 2 bonds here and 2 bonds there. These 2 hydrogens are bonded. Things like this. Okay. And then he says, well, we know, and this is one way to estimate entropy. So this is how you calculate entropy in physics of simple models. You say, well, we know there's this beautiful formula that the entropy is is the log of the number of configurations that the system can adopt, right? And there's a proportion, there's a form factor that maybe we should get rid of, but for some reason we all like, which is the Boltzmann constant. Okay, so you count the number of configurations, you obtain entropy with the Boltzmann constant. So all we have to do now is count. So if we know how many oxygens, carbons, hydrogens, nitrogens, phosphor, and sulfur there is in a bacteria, all we need to do is count the number of configurations, and that's it. And then we take the log, multiply by KB, we should get the right answer. I mean, he's a bit more refined than that. Okay, so he says, in fact, so that's the first thing. He says, so we could do this, right? We know that there's n, the total number of atoms, equals the number of carbons, plus the number of hydrogen, plus the number of oxygen, plus etc. Right? And so we can just say that the number of configurations, and this depending on your background may seem obvious or one of those combinatorics nightmare. The number of configurations that you can place the atoms is like this. And the idea is you can put the first atom whatever you want, right? So you have n possibilities for the first atom. Then, so this is factorial. Everybody's familiar with factorial? So n factorial is n times n minus 1 times n minus 2 and so on, right? So, okay, I'll just do a couple more minutes, okay? Right, so essentially you can say there's n atoms, right? And the first one has n places to be in. The second has n-1 places to be in. The third one, n-3 places. So these are all the different configurations that you can have. But you know that the carbons are indistinguishable. So you did some over counting, right? So you need to divide. Because the carbons, in fact, you can reshuffle them, and that doesn't mean anything, right? If you replace them with another carbon. So you need to put this factor in the denominator. Okay, is this something you're all familiar with? You know, probably depending a bit on your background, may seem a bit magical. Right? Okay, so it says this, yes. Yes. Don't care. It's a very, very crude estimate. Yeah. So all possible, you know, you can have all carbon, all hydrogen, that's fine. It'll be a rare configuration, right? But those configurations can happen. There's no, in some sense, normally, you know, you should be number of states compatible with your macro state, right? Here, there's no constraints. You know, say my macro state is just there's atoms. That's it. So it's like a gas of atoms, if you wish, right? The next thing that he says, let me see if I remember this, is he says, well, let's be a bit more refined than that and take into account that in fact, once you do that, there's multiple states compatible with each of those states, depending on how the bonds are placed. So say you see, that could be one arrangement, but I could also have something like this. Right? So you can change the bonds, and that's yet another configuration. So you need to estimate that. Right? So for a given placement of atoms, how many different ways you can place the bonds? Okay, and you need to do that. You need to give into account that the valence is 6. Right? So not valence. So V equals 6. Maybe Z is a better number. Okay. That means each atom can interact. Here you see 4, right? The one to the left, right, up, down, but in reality it's also back and forth because it's 3 dimensions and it's a cube. And so what he does afterwards, let's see. So the total number of configurations of the bonds would be how was this? I derived this a few days ago, but atoms, each element can. Right. So you have right. So you would say the number of bonds, the number of different ways in which the hydrogen can place its bonds, the first hydrogen, and the number of different ways in which the second hydrogen can place its bonds, times, et cetera, and then you get to so I shouldn't put right times right? And there's NH like this, right? So we're assuming all atoms can place the bonds in all ways. So it's unconstrained, so we're overcounting in some sense. And then we say times the number of different placements for the bonds of the first carbon, the second carbon, and so on, to the last carbon. And this will be NC, etc. And what we have here is something like this. Right? Bi times Ni. Okay. And so then we need to estimate in how many different ways can the carbon place its bonds, right? So carbon has one bond, right? So you can place it in 6 different ways. Sorry, hydrogen. So hydrogen has one bond, so it can place it in 6 different ways, right? And then carbon has 4. And so you can place the first one in 6 different, no, let me do oxygen. Oxygen is easier. Oxygen has 2, right? So then you can place them in 6 ways as this, right? You place this both here, both there, both up. So there's 6 such configurations. There's 6 on 2 configurations like this. Okay? And then you can do it for nitrogen in the same way. So it has 6 configurations where, for example, all the bonds, which has 3, go like this, plus 6 on 2 times 2, where they're like this, because this will be different than this, okay? Plus 6 on 3, and that gives you 56, okay? So very, okay. And you can do the same for the other atoms. And then you can multiply this weight by that factor. Okay, so these are the things that I can do in the Blackboard. Okay, and then you can plug this in this formula. Are there any questions so far? Maybe I'll just finish and you can ask me the questions afterwards. Anyways, only have one more second. Okay, so if you place this on this formula, then you do something that I mean, it may look like a very complicated formula and so on, but in fact, you can use the. So for those of you who know, you know, right? There's a Stirling approximation, blah blah, and for those of you who don't know, it's a very simple set of tricks by which this complicated thing, once you take the log, it becomes very simple. And essentially, what you have is, let me see. You can do something like KB. You have a term like n log n minus sum over I and I log ni plus sum over I and I log bi. Okay? And now we're in luck because we know this, we just calculated or show how to calculate these numbers. And this is pretty well known how many atoms there are in E. coli. And so you can put those numbers in, which I won't do. I won't show you how to do. And then you get a number. And the number that you get is 4, I'm sorry, that's a K, 4 joules per Kelvin per gram. And okay, so that's a number. And the one thing I would like to mention is that this number is roughly, so within a factor of 2 of the things that people actually measure by doing this very crude experiments. It was a pellet, they dry biomass, and they do the heat capacity and so on. Right? And any questions? So it's 10 to the 10, I would say, is it per cell? I think it's 10 to the 10 for E. coli, yeah.
The distribution of atoms is almost the same number of oxygens per, can we make that assumption here?
What assumption?
That the number of oxygens is the same as the number of hydrogens and such.
No, we're not doing that assumption. So we're keeping, that's why we have Ni.
Okay.
So actually 10 to the 10 is roughly the number of carbons per E. coli, right? And there's a different amount of hydrogen, carbon, and oxygen, and so on. But those numbers are relatively well known, I would say. So given everything that is unknown, I would say those are like the good numbers.
And can you tell us what paper is this exactly? Like
I don't know off the top of my head, but I can tell you the reference. It's a bit hard. I mean, it's an easy paper, but it's a bit hard to read. Like, many things of this person are a bit hard to read. Yeah, Surid.
Yeah.
So you said it sort of agrees with the heat capacity measurements from the dried biomass.
Yes.
Does it also agree when you account for hydration and things like that? So does it, what I'm asking is basically, does it agree with the living material cell?
Okay, so the question is, of course, nobody has measured directly the entropy of a living cell, right? What we can do is estimate the entropy of hydration. It seems to be a quite small number for what people report, like orders of magnitude lower, maybe 1%. Then there's of course the entropy of the fact that these atoms are not atoms, but then the proteins can be arranged in different ways and so on, right? So there's a very involved estimate of this in the book of Morovitz, which is not very user-friendly, I would say. And this is a bit slightly more user. At least, this after one afternoon or something, I could understand.
I'm trying to understand it a little bit conceptually. So that means that there's not a lot of difference that the fact that the bacterial cell exists in a certain structural way that it does? Or is there certain...
I think what this means, sorry, can you say that again?
Like in the sense that there's there's always, you have a fixed like macromolecular composition of a bacterial cell. It would basically have the same sort of entropic value if it was in a completely different having.
So, this is a good, so I guess the question is, how come this works if it's so rough? No, is this exactly. So I have to say I didn't have enough time to think very profoundly about it, right? So should this be an overestimate or an underestimate? I mean, in some sense, I guess you would expect this to be an overestimate, maybe, because you're counting too many configurations, many of which will not happen, right? So you're counting up, right? So I mean, it's very possible. I'm not making a strong claim about this. It's very possible that this is right for the wrong reason, right? I think many of these estimates may be right for the wrong reason. So like, okay, you're actually ignoring very important aspects, but you just threw in a bunch of numbers that are large enough that you got something kind of reasonable. I mean, after all, you need something like an n factorial in there on the size of the number of atoms. You need to take a log of that, and then you need to put the Boltzmann constant, and you should get something 1 order of magnitude up and down. I think that's why roughly you get something on a reasonable scale. I mean, the fact you get it within factor of 2 it's a bit mysterious, I would say. But when people say within factor of 2, it's not like the measurements are amazing either, right?
Sorry, Pablo, I missed the part. What exactly is the measurement that we're comparing this to?
So people do. So the measurements that people have done is measurements of the biomass entropy. So you mean what particular experiment people do?
Yes.
So the particular experiment people do is the dehydrate. So they go up, they lyse cells, they dehydrate them, they pellet them, so they form a condensed biomass chunk, cube. Sure. And then they cool it down, right? And then I think
they cool it down.
You cool it down, and then you measure the heat capacity, I think. So you increase the temperature little by little.
So you're just measuring the heat capacity.
Right, and then you're integrating that, and then you're saying the integral of the heat capacity should be it's pretty rough. If you know a better measurement.
No, I'm just curious what we're comparing this with. Okay.
So these are the kind of experiments. It's literally an attempt to directly measure the equilibrium entropy of biomass.
Bacterial material.
Yes. Is literally
heat capacity of.
Yes. There was another comment.
Yeah. So maybe I'm paraphrasing your question. I'm not sure. So if we contrast this to this picture of life is basically maintaining small compartments of lower entropy, right? By basically life means we increase the entropy in our surroundings, but maintain a lower entropy within cells or our body, then I think this shows that actually this reduction is small, right? So this in a way is a bit disappointing. It tells us that the living cell, the entropy of a living cell is not really strongly affected by the fact that they has to function and be alive, because none of, right, all this counting just takes the constituent components and the volume into account. There's no...
Well, it's a good point. I mean, I guess in some sense, I mean, this is not constrained by what you would say is the macro state of a bacteria, right? So this is not constrained by much of that order, other than the bacteria should have this component. So what you would expect is about the actual measurement to be lower.
Exactly, yeah. So I think that, right, so the true entropy should be lower because we know that, but this tells us there is not, that probably, since all these measurements agree, this maybe tells us that.
Well, I wouldn't go as far as all these measurements agree, right? I mean, there's like maybe 2 or 3 papers
yeah.
where people have measured these things, and they're not recent either. Nothing against old papers, just to say this is not something that people put a lot of importance to experimentally. So the
okay, right. So we could say that actually this heat capacity-based measurement also kind of
it
measures
destroyed
more the entropy of the constituent components.
Right, because in fact,
it retains more structure, but not that much more.
Exactly, because in fact, a lot of this large structure that you see is destroyed before those entropy measurements, right?
Yeah.
Like the protein folding entropy is destroyed. Yeah. You can look at the estimates of the Morovitz, right? When he has this book chapter, which I think one should just like sit and rewrite, you know. And there he would argue, I think, right? So the estimates are that even if you had included that, it wouldn't have been a huge difference, right? So I think what he says is that the main thing are the precursors, so amino acids and things like that. Once you have the bonds, then all the extra terms for folding and so on doesn't add. It's not a huge difference.
I had a doubt regarding the last formula. Why exactly is the middle term being subtracted from the?
What's the question? Your question is concerning where this formula comes from?
No, it's about like why are we subtracting the, the number of the sum of the total atoms?
Was there a minus here?
Yeah.
I mean, it comes out, right? But that's the cheap answer. The other answer is that because this is in the denominator.
Oh, okay.
So you have 3 terms, which are one is the log of this, one is the log of this, one is the log of this. This is in the denominator, so it has a minus sign. Okay, so maybe I'll leave it at that. We've already gone over time quite a bit with this question.
Sorry, just one question.
For me, it's fine, but
do we have an intuitive understanding of this magnitude in comparison to other things? Like
this what?
This magnitude of entropy. So
that's a very good point. So how, yes, that's the last half page that I have to say. So how does this magnitude entropy compare to other things? So I mean, you can take this type of number. You can maybe that's the last thing I will say. You could estimate the free energy of biomass by taking this number and is H minus Ts. No? Taking the enthalpy, the entropy, the temperature, and you get a value of the free energy of biomass. And you can then compare that to the free energy dissipation of a human, for example. And so in some sense, you can think about whether the free energy content of a kilo of biomass, how does that compare to the power dissipated of that same biomass, right? And what you get is that for the value of a human in about 1 day, it dissipates its own weight in free energy. So the free energy that biomass contains for a human, it takes about 1 day to be dissipated. For a bacteria, which is what we're, for microbes, in fact, is different. So what happens is that they dissipate more than what they contain. Right? So the microbes are very, very dissipative. There's a lot of free energy coming in and out, and very little stored. Humans, it takes 1 day, it seems.
But I mean, say what would be the entropy of soil or like the water? Is it
okay. So I don't know those numbers, but we can
much higher, I guess.
We should check them. Maybe let's talk during the poster session because probably people want to rest already. Okay. Thank you very much.